Hey guys, so we already talked about the genetics that we learned from Gregor Mendel, and obviously that was sort of the basis of where most of modern genetics is going to come from. Those are sort of the base rules, but we all know that that those are never the rules that apply in every case. And so what we're going to talk about today are a lot of cases that were outside of the things that Mendel studied. In a lot of ways, Mendel got kind of lucky that he was studying pea plants because everything did work pretty smoothly on the pea plants. And we're going to see that that's not the case in every situation. For instance, like this is a little diagram here from the Human Genome Project of human chromosome number 9. Human chromosome number 9 has 136 million base pairs. Okay, that's the pairs of DNA, the pairs of the nitrogenous bases that lock up in DNA. And there's a ton of different things that are coded for on this one gene, and of course we've got 23 pairs of these, and so on this one gene there's a ridiculous amount of information, and what's here is a bunch of things that can cause some problems, some sorts of defects. So there's a lot of stuff here, and so it's a little bit more complicated than what Mendel had originally thought. And so we're going to talk about some terms today that are what we call non-Mendelian genetics, or some things that were not those strict rules of independent assortment and dominant and recessive that Mendel talked about. So the first one we want to talk about today is incomplete dominance. And in incomplete dominance, what happens is that there's no real dominant gene. If we looked at this according to sort of classical Mendel genetics, what this would look like is that if we had a red rose and a white rose, that we would cross those, okay? And so we'd have our Punnett Square here. Sorry, me and the tablet are having some problems here. And so we'd split up our genes here, and so we've got r, and big R, and we cross those, and we would get hybrids. And in every case, according to classic Mendelian genetics, in every case, since we've got a big R in each case, we would get a red rose out. And then, of course, we could breed them again, and then we would get some white roses out. But what researchers found is that that's not always the case, that sometimes when you combine a red and a white rose, that what you actually get is a pink rose. Now that kind of makes good common sense to us. And the reason behind that is something called incomplete dominance. And basically what it is that we blend these two things together, and instead of us getting one thing being dominant over the other, instead both traits sort of come through. And so you get a little bit of red and you get a little bit of white, and it's blended together to give us a combination. And this is called incomplete dominance. Something that's pretty similar to this is something called codominance. And in the case of codominance, what happens is that they don't really blend together, but rather different parts of the body or different parts of that organism are going to display those things in different places. So for instance, you might get a rose that has red splotches and white splotches on it. Or another really classic example that we see all the time, this is like the common classic genetics example, is a roan cow. And so you've got red fur and white fur, but it's in splotches all throughout. And in fact, if you even looked in the red part, you would see a little mix of each, and obviously you can see a little mix of each here. So it's not like it was a blend of the color to get a lighter color that's both of them, but instead they're each displayed in different areas. And remember, that's the phenotype, okay? The phenotype, of course, is what we can actually see. Another example of something that was definitely beyond what Mendel did at the time is multiple alleles. So normally when we do our genetic stuff, we've got alleles of stuff, and so, you know, for instance, you might have an A and a little a. What happens in the case of something like blood types is that we've actually got an A and a B, and in fact an O, and so you've got different alleles that are going to come in here. And so they're not the same thing, okay? You don't just have an A and a little a, you've got A and B, and so those are going to lead to all kinds of different things. And blood typing is super interesting. Of course, we've got the separate video that you guys have already watched on that. So that's a case of multiple alleles. So it's not, in this case, it's not multiple genes, it's just multiple alleles of that particular gene. So it's still one gene, but with different ways that it can be expressed. This all leads us to a guy named Thomas Hunt Morgan. And Thomas Hunt Morgan did massive studies on Drosophila. Drosophila are basically fruit flies, and they're sort of the classic genetics thing to work with because their life cycle is really quick, and so you can breed them and then get the next generation and then the generation after that really quickly. They're really easy to work with, and then of course no one really cares that much about euthanizing some flies for the most part. So Thomas Hunt Morgan is going to do all of these studies, and what he does is that he agrees with a lot of Mendel's stuff, but he also finds cases where his experiments are not working out according to the classical Mendelian genetics. So a couple of good examples of that. Gene linkage is a great example. What Morgan found is that genes don't always independently assort. Okay, we've sort of operated under the assumption that, like, if we were doing our pea plants back in our Mendel's genetics, so we were saying, well, okay, let's say that we've got yellow peas or green peas, and then we've got height of the plant, and we would say that those things independently assorted, so that you would get a Y and you would get a T, and that those things would come separately and you could split them off. But what Thomas Hunt Morgan is saying is that that's not always the case, that sometimes these things are located on the same gene. And so a classic example of this is that something like red hair and freckles. It's really common that you see someone with red hair that also has freckles on their skin. And so the reasoning behind that is that those two things are coded, okay, that the gene that expresses those is on the same chromosome. And so therefore, most of the time when those things get split up during mitosis and meiosis, when those things get split up and sorted, that they're on the same gene. And so the only way that they're really going to get separated, the only way that you're going to get, say, red hair without freckles, is if crossing over happens and those two things get separated off on the gene. So remember, we pulled off one part of the gene and it went to a separate part. And so that might be a case where those things get split up. But it doesn't happen as much. In fact, what Thomas Hunt Morgan found was that he could tell by the percentages of his phenotypes how close together on the same gene these two things were. So, like, let's say that, you know, if freckles was over here, then he would see those things split up a lot more commonly. If, on the other hand, he always found red hair and freckles together, they must be close together on the gene because they're not getting split up during that crossing over that happens in meiosis. Okay, so that's gene linkage. That's when genes are on the same chromosome, and so therefore they're not going to get independently assorted necessarily. Another really common thing, and this is really interesting, and this is really useful to us in real life because we've been treating everything so far as though there were one gene that coded for it. So like there was a gene for eye color, there was a gene for height, or there was a gene for the color of your skin. And really, in the real world, that's not the way it works. And most traits that we get are what we call polygenic. Which means that multiple genes are going to code for that one single trait. Sort of the really good example of that would be something like height. I mean, height is obvious that it's not like you just have tall people and short people, and all tall people are the same height, and all short people are the same height. And there are cases where you can have two relatively short parents that have really tall children, or really tall parents that have really short children. And that shouldn't be the case according to Mendel's normal dominant recessive independent assortment, and one gene codes for one trait. What actually happens is instead that we're going to have multiple genes coding for multiple traits. Okay, and that's pretty important. Sorry guys, I had to click the charger thing here. So let's, for instance, take height, for example. So there's a variety of different heights that you could be. And so let's say then that there's 3 different genes that code for height. Let's say that there's an A, a B, and a C gene. And let's say that you're a sort of in the middle person, okay? So you've got 3 genes that code for height, and that they're each coding that you're going to end up in the middle, okay? So you've got a dominant and a recessive for each one of those genes. So that means that your genotype would be AA, BB, CC. And let's say that your phenotype, let's just give a number to that phenotype. Let's say that you're height 3, okay? Why 3? Well, let's go down here and let's say that there's a 0 and a 1, a 2, a 3, which is what you are, 4, a 5, and a 6, okay? So let's say then that you mate and have children with another person that's a 3, okay? So there's 2 of you, and you both have that same genotype, and therefore that same phenotype in that case, and so you're both 3s. Well, does that mean that all of your children are going to be 3s? Well, definitely not. In fact, you can already tell that we're going to have a really complicated Punnett Square for this, right? Because we've got 3 actual genes here. Okay, we've got an A gene, a B gene, and a C gene. And so if we set up a Punnett Square, it's actually going to look like this. Now this looks incredibly complicated, and actually I made it a little bit easier because in the boxes I didn't actually put the genotype, I just put the phenotype there, okay? And so what this means is that if you had 2, say, average height people or 3 height people, that they can actually have children in any of the height categories that we talked about there. So they can have really short children with a 0. They can have really super tall children, MDA style, that have the 6 there. Now, that's not likely. The probability is pretty low. You can see that there's a 1 in 64 chance there, which is about a 1.5, I want to say like a 1.6% chance. And that most of their children, okay, the greatest probability is that they're going to be in the 3s. And then there's a slightly less probability that they're going to be in the 4s or the 2s. And really, if you consider all of that, all of those categories there, okay, the greatest probability is that they're going to end up somewhere in there. So maybe a little bit higher than average, maybe a little bit shorter than average, but probably somewhere around the average height. That's about 50 out of your 64 chances. Now, what's kind of interesting is that what Thomas Hunt Morgan found is when he did these studies is that corresponds to what we know as the bell curve. Okay? Whoops. I totally just threw that in the table. That what he found is that most cases you get an average distribution, that most people are going to end up in the middle. But you can have outliers there. And he found all of that by working on his flies, that he found that most cases he would get what he expected, the average values, but you can still get these outliers out there, and that they weren't in the normal Mendel calculations for having one gene, that there had to be multiple genes that were coding for this thing. And there are a lot of traits for us as humans that applies to. So things like eye color, things like skin color, things like height, to some degree your body style or your weight plays a role in that. And there are multiple genes that code for that. It's not just one thing. And that's really good news for, say, my son, who I've been telling him he's going to be short because his daddy's average height and his mother's short, so he's going to be short. Well, obviously, that's not necessarily the case. It is likely, okay, it's really probable that he's probably going to be short or average at best. But it could be that he happened to get those multiple genes that when they independently sorted, that he got the ones that really gave him this 6 value. Not going to happen. I can kind of tell that already looking at it. But it's possible. And that's what multiple genes allow. It means that you don't necessarily have the exact same traits that your parents have because there are multiple genes that code for that. So it's a little bit more complicated than that. All right. So the last sort of special case we want to talk about here are sex-linked traits. And what sex-linked traits are, basically it just means that these are traits that are carried on the X chromosome. So X and Y chromosomes are what determine whether or not you're male or female. Okay? If you get 2 X's, then you're female. If you get an X and a Y, then you're male. That's sort of what determines gender from a genetic standpoint. It's kind of interesting because you can only get the Y chromosome from the father. So it sort of used to be back in medieval times that like kings would like say in the case of Henry VIII, that he would like divorce his wives or behead them or whatever because they weren't giving him sons. In reality, it was all his fault. The women don't have anything to do with the gender determination. The gender determination is completely comes from the guy because the guy is the only one that has a Y chromosome. The woman can't give a Y chromosome. She's only got X chromosomes. So sex-linked traits are basically traits that exist on the X chromosome. And so what that means is that your gender is actually going to determine a lot about how these traits actually get expressed. So I'm going to pick one that for me obviously is pretty personal. I'm going to pick baldness. Now baldness is actually a little bit more complicated than this. There are multiple genes that code for it, but the main expression of it is sex-linked, okay? And that means that there's going to be a little marker on the X chromosome, and I'm just going to call it X sub B, that codes for baldness. And so the way this works is that, let's say that you have a father who is not bald, okay, normal XY father. And then you've got a mother who is also not bald, but she's carrying the gene. She is what we call a carrier female. And I've got that arrow there, but it also goes there. You'll see why in a second here. It's going to come up. She's what we call a carrier female. She's carrying the gene, but she's not expressing it. She doesn't appear bald, but she has one copy of the bald allele, okay, one bald allele. So let's do our cross here. So if we do the first, if we're just going to do our regular Punnett Square crosses, bring down all the female stuff, okay, so all the X's, and then we're going to bring across all of the male chromosomes, so the X's and the Y's come across. then this is what we get. Now, obviously, see, first off, there's a 50-50 chance of getting a male or a female, okay, each time. But remember, that's each time. It doesn't mean that, like, if you have 2 kids, you're going to get a male and a female. I obviously, again, am a good example of that. I've got 3 kids, and they're all boys, okay? So, and again, that's all my fault. I only gave Y chromosomes, okay? So how this relates to sex-linked traits is by this. So we do the cross, and what you see here is that you've got the case of you've got percentage-wise, 25% chance of being a carrier female, 25% chance of being a regular female, 25% chance of being a regular male, and then, all right, sorry guys, having a problem finding where my, and then a 25% chance of being a bald dude, of being me, of this mess up here, is because the carrier female, okay, in this case my mom, okay, passed down an XB gene, and I happen to get that, and then the Y chromosome comes from my dad. Now here's the deal. Normally, baldness would be recessive. So if you look at the female up here, XB and X, my mom wasn't bald, okay, which means that baldness is a recessive trait. But here's the bad news for guys with these sex-linked traits. Guys only have 1 X chromosome. And so if you get that passed down from a carrier female, then you pretty much automatically get that trait, okay? So I'm bald, for instance. Because I've got that trait. I had a pretty good chance of getting that trait. Now, I didn't have to get it. I could have lucked out and got this one instead, but this is obviously my genetic code. There are a couple of other traits that are really common for that. Hemophilia and color blindness are also really common sex-linked traits. And again, the gene for it, okay, actually comes from the mother because the father is giving the Y chromosome. And so therefore, he's not actually giving the trait for that particular disease. But what is happening is that since the father gives the Y, is that you only get the X and the Y. Now, what will happen genetically for the other traits? This carrier female, still not going to be bald. Okay? The only way that you would get a bald female, okay, and this is going to be much more likely, much more unlikely, is that if you also had the trait on the male. Okay? So you had a carrier dad and a carrier mom, and then in that case, when you did that cross, you would get this, okay? And so actually, those B's, hold on, those B's actually should be, yeah, okay, that B there should be over there. And so this female, lucky, carrier female, carrying the gene, but not expressing it, not bald. But this one is. Now, the good news is it's pretty unlikely still for females. It's about 1 in 100 cases do they get a gene to be genetically bald. For men, though, about 1 in 10 cases, it turns out to be that way. And the same thing is true for stuff like hemophilia and color blindness. There are colorblind women, but they're much less likely. There are hemophiliac women, but again, much less likely. And again, it's not just the sex-linked thing. There's multiple genes coming in here, but that's the main thrust of it, is that it's going to come from the X chromosome here. So we'll talk a little bit more about this in class, but that's the basic gist of all of this non-Mendelian stuff. So you need to sort of go through each thing and know what an example of each is, because that's